Hi, this is Mrs. Sloan, and yeah, I just got out of the pool. I know, look and fly. We are going to continue on chapter one. We have already done chapter one, part one, and in that we looked at the beginnings. We looked at the characteristics of life, and we took a deep dive there. Then we transitioned into the classification of life, and here in part two, what we're going to focus on is we're going to look at the biosphere and kind of break the biosphere down, and that will be a review for you. And it'll be a review because we started off our school year talking about ecology. And this is chapter one, who's giving you kind of an overview of ecology. We've already had a deep dive on it. So that will be a review for you. And then we're going to go into the process of science and the scientific method and take a little dive into using math to analyze data. And then we will end with science and social responsibility. All right, so I'm going to make myself a little bit smaller here and skip down this bit we have already done. So we are right here, and I want to present. So if you remember from the part one, we did how is life organized, right? Living things are organized, made of cells. We said they acquire materials and energy. Remember that? They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. So we went over those. And then, but the very first one is living things are organized, made of cells. Let's review how it is organized. It goes atoms, right? Build molecules. Molecules build organelles in eukaryotic cells. Organelles build cells. That's where life starts. Then you have tissues, organs, organ systems, and organisms. Okay, and those are species, right? And then you have groups of the same species. That's a population. Multiple populations is a community. You start taking into consideration the environment, temperature, right? pH of water, salt, salinity, right? That's your ecosystem. And then the biosphere is where all life exists. And in part two, that's where we are starting talking about the biosphere. So on your notes, you already have this, where all living things are found in the air, in land, water, right? That's what the biosphere is. Then this next slide, I really like it because it should be a review for you. From what we've already talked about. So ecology is looking at the relationships between living organisms and their environment, right? This part right here, and their environment. And we know that populations make up communities. We know an ecosystem is the community and all of its abiotic environment. A habitat is where you live. Remember, a niche is what you do. You have those on your notes already, what a community is, all the different populations of one area, and an ecosystem is the community plus the physical environment. Okay, this next part is also a review. You remember that chemicals cycle and energy flows, and you should remember your different cycles. We talked about the water cycle, we talked about the phosphorus cycle, we've talked about nitrogen, we talked about carbon, right? And we could see how energy moves through living organisms. We talked about trophic levels, yes? So on chemical cycling and energy flow, you have everything for that. Chemicals move through living organisms and are broken back down to inorganic molecules, whereas energy starting from the sun flows through organisms and eventually dissipates as heat. And in a later unit, we'll talk about the different laws of thermodynamics and how specifically that moves through autotrophs to heterotrophs. Okay, then you can't talk about ecosystems unless you talk about how man, here's Farmer Ted, he looks like an organic farmer, seems like he'd be a nice guy, but remember, man, when we jump into ecosystems, we usually modify them for our own purposes. So humans tend to modify ecosystems for their own purposes, oftentimes decreasing biodiversity. Okay, put a pin in biodiversity. I'm going to come back to that. I just want to mention the two really threatened ecosystems on our planet, and that is rainforests, right? And then the rainforests of the ocean, those are coral reefs. They have a lot of diversity. It's critical that we maintain that biodiversity. You do not know, if you want to be like just super selfish human terms, there could be some organism that lives within one of those areas where we have the greatest number of species, the greatest amount of biodiversity that could save your life. It could be the drug that you need to save your life at some point is found in one of those organisms. So when we talk about biodiversity, you're talking about all sorts of ecosystems, and we will study those at our final unit and the key parts of what kind of things we do as humans that impacts those. We will revisit ecology at the end, but just to give you an overview of what biodiversity is, you're talking about how many different kinds of species you have out there. You're talking about the diversity in their genes of the different species that they have, and then ecosystem biodiversity. Because we tend to go in and change it. I mean, if you look at the area we live, my students live in Oak Park. And if you look at Oak Park, it's a chaparral environment. But if you drive through it right now, through Oak Park, it doesn't look like a chaparral environment, except for up in the hills. But around people's homes, it looks like maybe a tropical area or a coniferous forest, because we have changed that ecosystem to what we want around us. So we'll talk about man's role, biodiversity. We'll revisit that at the end. All right. Okay, but that should all have been a review for you. So next we want to move into the process of science. And then little quote there, okay? Then look at this little cartoon. I know it's simple, but it says a lot. So somebody's taken a look at this tree or whatever, and they might make some observations that they see. They might start asking questions. That's what number 2 would be, right? Asking questions about what they've seen and do some research on that. Once they've done some research, that's why they say hypothesis is an educated guess. It's not like you're just pulling it out of a hat. You're actually making observations, thinking about it, doing some research, and then putting forth your hypothesis. Now, we will talk about 2 different types of hypotheses today in a little deep dive in a little bit, okay? Then you're going to do your experiment. It's going to be a controlled experiment, a repeatable experiment. You're going to analyze your data, and then you'll make some conclusions. So here is all of that uncovered, okay? And you have all those steps in your notes. A couple of things I would point out on your analysis right here. We really focus on claim, evidence, reason. If you're going to make a claim, you're going to back it up from evidence from your experiment, and then try to explain it and have that reason for it. And that's what our deep dive will be today, is how can you know if something is significant or not within your data when you're analyzing it? All of that is in your notes already. Okay, so just take a minute, look at this. This should be a review. Making observations, thinking of interesting questions, you're formulating your hypothesis, you're testing your prediction, gathering data, you're analyzing that data. You may need to refine or alter and experiment again. Potentially several different repeated experiments that are supported could lead to a theory. All right, so one issue to look at is variables. So you have your experimental or independent variable, and then you have your response or dependent variable. So your experimental variable is your experiment. It's what you're varying. Maybe you're concerned about artificial sweeteners and cancer, being linked to cancer of the bladder or something like that. So you might do an experiment on mice. Now, that's another whole issue we can talk about. Please don't get upset about that. That does occur, but that's not what I'm talking about here. But you may do varying amounts of sweetener, and then your response variable, your dependent variable, would be the number of tumors that you find in rats as a result of that. That's your data. So your response or dependent variable, and the way I remember that, like if you think dependent variable, this is your data, right? Dependent and data, it's your response. Your experimental or independent variable is what you chose. So for instance, if I said, I wonder how light affects plant growth. That's like a 3rd grade experiment, right? Your varying amounts of light that you provide different plants would be your experimental variable or independent variable. How the height of the plants that they grew underneath those different conditions, that would be your response or your dependent variable. All right. So here we want the kind of data that you would collect. When I talked to you about the experiment we looked at with rats and sweetener, the quantitative data would be the data that you could graph. That's the numbers over here on the right, okay, is the type of data you could graph. Qualitative data would be like the rats didn't look healthy, they weren't very energetic. You can't graph that, right? That's very descriptive. But quantitative data is dealing with numbers and measurements. If we were looking at how light affected plants, if you were giving me the height of the plant, that would be a quantitative data. If you said the plants kind of looked brown or dried out, that would be qualitative data. And again, you have different types of quantitative data: discrete, there were 3, there were 4, and then continuous would be over a set of data, right? Like a line graph, all right? Now, the key to knowing whether your data is significant or not when you're analyzing it is about using mathematics to analyze your data, right? So, for instance, if I said, hey, this is a very simplified thing. If I said, flip a coin 10 times, if you were going to predict, how many times would you predict if you flipped a coin 10 times that you would get heads? Okay, hopefully you told me 5, right? Because you know it's either heads or tails. So if I told you that I got heads 6 times and tails 4 times, would you be concerned about the coin? Okay, what if I said 7 and 3? What if I said 8 and 2? At what point do you think maybe there's something wrong with that coin, right? So we can't just do it on feeling. You have to use math, right, to analyze your data to know if whether it's significant or not. So that's where we're going to head mostly in this video today. So how do you know if your data supports your hypothesis? When are the numbers significant? How do you know whether to accept or reject your hypothesis? And what types of hypotheses are out there? So for those of you who are using my notes, you have science practices NGSS, and you have the different science practices. And let me go back real quick and fill those in for you. So number 1 was asking questions. Number 2, developing and using models. Number 3 is planning and carrying out investigations. 4 is analyzing and interpreting data. 5 is using mathematics and computational thinking. We're going to focus on that. 6 is constructing explanations. 7 is engaging in argument from evidence, claim, evidence, reason. And 8 is obtaining, evaluating, and communicating your information. Now, just to hit a return a couple of times underneath that and take notes, okay, on what I'm going to give you, because I don't have any additional notes for you in there, but I want you to add it underneath science practices NGSS. So just hit the return there, and I'll tell you some key parts to write down, okay? So we're using math to analyze data, and what we're going to be doing is I'm going to teach you the ChiSquare test. And in order to understand that, we need to talk about the different types of hypotheses. And so I'm getting you to that point. Remember, we already talked about independent versus dependent variables. Independent variable is what you're changing, the amount of artificial sweetener, the amount of light. The dependent variable is your data. How many tumors were found in the rats? How tall did your plants grow? Okay, so that's our experiment. Now, when we write a hypothesis for that experiment, there are 2 different hypotheses you can have. So in your group shared notes, start with 2 types of hypotheses, right? 2 types of hypotheses. So the first one, let me make myself smaller here, okay? And you don't need to type all of this out. You'll see what I mean in just a minute, okay? A research hypothesis is one that you are very familiar with, like if, then, because. If I do this, then this will happen, and here I'll tell you why it will happen. It is saying A is connected to B. So for instance, I would say the more light I have, then the plant should grow better. I am saying there's a connection between A and B. If I give more sweetener, artificial sweetener, as I increase the sweetener, I'm going to increase the amount of tumors that I would find in my rats, all right? That is a research hypothesis. A is connected to B. So on your notes, research hypothesis, A is connected to B. Research hypothesis, A is connected to B. A null hypothesis says there is no connection, that there is no connection between A and B. and A does not have an impact on B. And you're like, why would you say that? I'm going to show you. Just believe me. Just trust me on this, okay? This would be saying that the amount of sweetener I give the rat will not impact the number of tumors it has. The amount of light that I give that plant will not impact how high it grows. The amount of yeast I put in my bread dough will not affect how high the bread grows, okay, or rises, okay? There is no impact. And I will tell you why you would want to use a null hypothesis, okay? So take a look right here. A research hypothesis is called an H1, okay? So that is saying there is a connection between A and B. A null 0, is an H0. There is no connection between A and B. Okay. Now, here's why you would want to do it. I wonder, okay, this is my driving question, is there a connection between the amount of sleep you get and your grades? Is there a connection between the amount of sleep you get and your grades? Now, let me talk to you just for a minute up close, okay? Do you think intuitively that there's a connection between how well a student does over the course of the year if they have regular sleep versus very small amounts of sleep? Probably there might be a connection. But what else affects your grades in a class, right? Whether you connect with the teacher or not, whether you've heard the material before, maybe what time of day you go to that class, whether you like that material or not, right? There's a lot of factors that can affect your grade in a class, and it's just not the amount of sleep, right? How difficult is the material? If you took a class on 3rd grade math, I bet all of you would get A pluses in it because you already understand 3rd grade math, right? If I gave you a college level course math, maybe you would struggle with that a little bit. I don't know, right? It depends on what your experiences are. So the reason why people use null hypothesis in experiments is because if you say there's no connection between A and B, but yet you have overwhelming data to say maybe there is a connection, then this supports that there is a connection between A and B. You're kind of backdooring it, going about it the same way. Because I can't say it's just sleep. There's a lot of things that impact the grade. So can I say that sleep is one of the things that impacts your grade? Let me show you right here. Okay, so if students get at least 7 hours of sleep a night, then they will earn higher grades. That would be a research hypothesis, if then, okay? But that's not always true, right? You could get 7 hours of sleep, but if it's really, really hard material and you're not interested, you're probably still not going to get a higher grade. So it's better to say there is no connection between the amount of sleep a student gets and their grades, and then see if the data disproves that, okay? At a significant number, and that's why you need your ChiSquare test. All right, so chance is always going to play a role in any experiment, right? So back to flipping the coin. If I flip the coin 10 times, you would predict 5 and 5. That seems reasonable. But it could also go 6 and 4. That's chance. So when you look at chance, it's like how much of it is acceptable. And where scientists draw the line again and again and again is if you get this data only 5% of the time or less, then you're going to have to say that's probably not due to chance. It's due to something else. It's due to a connection. Okay. So the null hypothesis would say your data that you get, it's just chance, right? If you get better grades, if you're sleeping more. However, if that is only likely to happen 5% of the time statistically or less, then maybe there is a connection between sleep and grades. And if you disprove your null, if you can't support your null, then you end up supporting the alternate or research hypothesis, which makes the connection. So by disproving your null, it will end up supporting the alternate. If I say there's no connection between the amount of sweetener and tumors, but then I get overwhelming data that says this would only have happened 5% of the time, then I'm going to go, okay. So I disprove the null and I say there is some sort of connection between sweetener, right, and tumors that you would have. All right, so let's put this, let's use something simple, okay? Let's use a deck of cards, okay? Keep your large mammalian brain going here. If I were to shuffle a deck of cards, and I told you I just bought this deck, maybe I'm lying, I don't know. I just shuffle this deck of cards, and I deal out 20 cards. Intuitively speaking, you would say, well, I bet that 10 of them would turn out red and 10 of them would turn out black because half the cards are red and half the cards are black, right? But what if instead of getting 10 and 10, I get 8 and 12? Does that mean I did something funny to the deck of cards? Am I trying to cheat somehow with the deck of cards if 8 were red and 12 is black? Is this significant? Well, let's look at the data. Let's look how a scientist would approach this. Okay, so I observed, okay, that there were 8 red and 12 black. How many did I expect? I expected 10 red and 10 black. How much was I off, right? If I observed 8, but I expected 10, how much was I off? Well, 8 minus 10 is 2. I was off by 2, right? 12 minus 10 is 2. I was off by a fact. If it wasn't red, it was black. That was my only choice there, right? So they were both off by 2. So what you do is you take the difference that you got from what you expected, okay? So 8 minus 10 is minus 2. 12 minus 10 is plus 2. And all you do is you take the difference from what you expected and you square it and put it over what you expected. So minus 2 is 4. 4 over 10, right? 0.4. 2 squared, it's going to be the same number, right? Because if it wasn't red, it was black. So that's 4. 4 over 10, 0.4. 0.4 plus 0.4 equals 0.8. My friends, if you don't understand what I just did right there, that is the chi-square test. You take the difference between what you observed and what you expected, you square it, put it over what you expected, and add it up, okay? And that is your chi-square value. Now, great, 0.8, what does that mean? All right, there's a table that you can use to analyze 0.8, okay? So I just want to show you the math that we just did. Observe minus expected squared over what we expected for both red and black. Okay, when you say your chi-squared number equals 0.8, don't then square root it. This is a thing. It's the chi-squared number. Okay, so it's 0.8. So now all we need to do is look it up on a chart and see if 0.8 is a big deal or not. So what we're going to be looking for on our chart is that it's a big deal if it would only happen 5% of the time or less. Meaning, like, odds are that you would get 8 and 12 is only 5% of the time or less. Then we're going to say there's something wrong with the deck of cards. But if it would happen more than 5% of the time, then we could say it's just due to chance. All right, so here's that table. Now, the next thing I have to teach you is degrees of freedom. Okay, so far what you have in your notes is you have the 2 types of hypotheses, right? The next thing I want you to add in your notes is degrees of freedom. Degrees of freedom is always equal to 1 less than the number of choices you have. Degrees of freedom is 1 less than the number of choices you have. Well, how many choices did we have? Red and black, right? So if you weren't red, then you had to be what? Black. So you had 1 degree of freedom. Okay? If I was talking about all 4 suits of cards, right? You have clubs, diamonds, hearts, spades. Then my degrees of freedom, 1 less than the number of choices I have, would be 3. Okay, that's how you find the degrees of freedom. Now, if you look right here, let's go back to our chi-squared value of 0.8. So our degree of freedom, because if we weren't red, we were black, right? We have, take away 1, we have 1 degree of freedom. So, skip to a scary chart. Look right here. Okay, let's find 0.8. Okay, let's find, where is 0.8? 0.8 would fall somewhere between 1.32 and 0.46. Okay, that means, let's look at the probability here. This is a probability table. We would have expected to have gotten 8 and 12 between 25 and 50% of the time. So yeah, we can accept that data. 8 and 12, yeah, that's okay. I would predict that would happen between 25 and 50% of the time. Now, I want you to observe something as you work from left to right. What happens to the chi-square numbers as you work from left to right? Do they get bigger or do they get smaller? Okay, what did they get? They got bigger. Now, remember how we calculated the chi-square number. We said, what did I observe? What did I expect? What was the difference? I squared it and put it over what I expected. If that number is big, that means I was off by a lot. If that number is small, it means I was off by a little, right? So look over here. To have a chi-square value of 0.016, well, that could happen 90% of the time. That's very predictable, right? But if I got over here, if my chi-squared number was 5, if my chi-squared number was 5, that's in the reject zone. That means it would only happen between 0.1 and between 1 and 5%. Nah, that's not due to chance. Something came to play there. Something came to play. Now, this is a smaller chi-square test, but if you look, a smaller probability table. This is a larger one. This takes you up to 10 degrees of freedom, okay? But it doesn't matter. Even in this larger chart, not intimidating, our chi-squared value was 0.8. So if you look right here, this particular chart broke it down even more. So 0.8 falls in this bigger chart, you can see between 30 and 50% of the time. So that is acceptable. Here you can review again. Degrees of freedom is the number of categories we have minus 1. Okay, big breath in, let it out. Our deck of cards was fine. Okay, let's try another one. All right. Oh, here, let me show you this. Now I'm going to show you the actual formula for this, which looks more intimidating than what it was. Okay, so literally all your data that you're going to analyze with all of your experimentation, which would be super awesome if we ever get back into class, but we're going to try it with M&Ms and see how you do, is with our expected values there. M&M colors, it's a fun lab. And what all this means is this: take what you observed, right, minus what you expected, square it, put it over your expected, and you do that for all the different choices that we have. And that's what it means, just sum of. Okay. When you're seeing this stuff here about phenotype, that's because we'll use this in our genetics units as well. So that's our chi-squared value. It's to analyze whether our data falls within the realm of possibilities, just variation due to chance, or do we need to consider something else? All right. So let's try it again. Okay. Let's try it again. Let's use something else. Now, this is from 2016 data. Yes, 2016 data. When they went out and surveyed how many professional jobs where you would be a chemist, how many were men and women, they found that 35.2% of all chemists were women. Okay. Now, when you do special hugs and make a baby, there's a 50-50 chance whether you're going to make a biological boy or a biological girl, right? So we would think, if sex didn't have anything to do with job choice, we would predict there should be 50% of the chemists should be men and 50% in a binary world, and 50% should be women, right? 50-50 in a binary world. But here it finds that 35.2% of chemists are women. Is that significant? Well, to write a null hypothesis, right? To write a null hypothesis, you would say there's no connection between the field of chemistry and the sex that you are. It's not influencing your job choice. Okay, so you can see this research hypothesis, this null hypothesis says women are not underrepresented in the field of chemistry. This could be just due to chance. 35.2% instead of 50% of chemists are women. Well, let's do the math and see if that is a statistically significant number. So we observed 35.2 are women. So what does that say? How many of them then are men? You just take 100 minus 35.2, right? So I know this is very sloppy, but I observed 35.2 female, and that means there would be 64.8 of the males then, 64.8 of the chemists are males. So what would I expect? Well, I'd expect 50-50 because that's what it is on our planet, right? So how much am I off? And I want to remind you, whenever you use percentages, do not do it as a decimal. Do it as 35, not 0.35. Otherwise, it's not going to work for you. Okay? So how much are they off? What is the difference? Well, I would just take 35.2 minus 50, remember, and square that and put it over what I expected. So let's take a look right here. Okay, so I made a little chart for you. So here's what I observed. Here's what I expected. So try this. Just pause the video for a minute and see if you can do this on your own, right? This is great. This will always help you to be able to analyze your data. So I take what I observed minus what I expected, get that difference. Then square that difference and put it over what you expected. Okay, do the same thing over here. So try that for both and see if you can get those two numbers. Then we're just going to add them together, and we're going to look on our probability table. All right, pause if you need to. Pause. Okay, here we go. Because now I'm going to show you the answer. Okay, so here is the difference. We were off by 14.8%. If I wasn't male, I was female, so there's not a lot of options there. You square it, 219. I put it over what I expected, 219 over 50 gives me 4.4 and 4.4. Now I just need to add those up. 4.4 plus 4.4 is going to be what? 8.8. Let's go see if this is significant when we look on our tables. Okay, so here it is, all done. Remember, I would have my degree of freedom would be 1, because if I wasn't male, I was female, right? So my degree of freedom is 1. So let's go look on that ChiSquare test. All right, so degrees of freedom, I'm looking for 8.8. I'm looking for 8.8. I'm reading from left to right. Here's 3.84. Here's 6.64. 8.8 is bigger than 6.64. So that means there's less than a 1% chance that would happen just due to chance, right? So that tells me something, right? My null says there's no connection between whether you're male or female in your job choice. That's what the null hypothesis would say. But to get only 35% of chemists being women, there's a less than 1% chance of that ever occurring, just due to chance. So now we can reject that null hypothesis, right? Because job choice apparently has a lot to do with whether you're male or female. Apparently, women are not choosing to be chemists. Now, we can open that up and say, why is that? Why are women not choosing to be chemists? But that would be another day. Right now, we can say it is statistically significant, and we can say by rejecting the null, by saying there's a 1% chance, we're rejecting this, this is not statistically likely to happen. Now we can support the alternative hypothesis that says there must be a connection between what sex you are and what job you choose, at least for chemistry. Okay, I know that was a lot. But I needed to move this part forward. Normally, we would talk about this when we do our behavior lab at the end of the unit. But as I now know, as of this last week, that we are not coming back to campus, I needed to move this up so we could use it on some earlier data. All right. Plus, you kind of have a slow week this week. So now, I will revisit that in class with you. All right. So now let's talk about science and social responsibility, the last tail end of your notes here. All right. So as far as, in this day and age, it's crazy that I'm even having this conversation. Science has to analyze data, and you have to decide whether it's statistically significant or not, like the number of fires we have in California right now, right? And there's a water budget. If you're wondering what's going on, when it's hotter, more water is going to evaporate out of the plant material, which makes it drier. The plant material is drier, and then fires will spread more readily. Then when you have all that water, right, we have a water budget. So the water on this planet, right, is finite. So if that water is leaving plants, guess what? It's going down in the southeastern part of the United States, and it's coming down, right? We're having tornadoes and floods, right? They're having that extra water and all those storms because we have less water here in California. So do we have any responsibility as scientists to convey this information so you can make good choices about policies that we enact and who we elect to enact those policies, right? So technology is the application of scientific knowledge for a practical purpose. So what ability do we have to measure temperature over time, to measure ice fields over time, to measure drought or rainfall over time? So technology is the application of scientific knowledge for practical purpose. Science is not a political party. Science does not make value judgments. It gives you the data, right? And then as a society, we have to say, what are you going to do about this data? I guess you could choose to ignore it. Certainly people do. They'll just say, no, not true. You now know how to analyze data, right? And there is truth that is provided through that data. Your interpretation is not open for interpretation in that, right? We, in fact, are warmer. So what are you going to do with that data? Okay, so science does not make value judgments, right or wrong. It's not making value judgments. This is what we have to do as a society. How are we going to use the technology? How are we going to use the information that we have? Now, there's ethics like that, like I can apply to climate, but let me give you another one that's ethics. For instance, we have the technology, if a child is born early, right? So when a child is full term, let's say 37 weeks development or later, and they are born full term, what if you can save a child that's born at 30 weeks? Do you do it? What about 25 weeks? What about 20 weeks, right? If you have the ability to do something, should you do it, right? If somebody is in a coma and you could keep them alive for 50 years because you have the ability to help them maintain homeostasis, should we do that, right? So just because we can do something with our scientific knowledge, should we do that? So that's part of what society has to do. So science does not make value judgments. Moral and ethical decisions must be made by all people. But it's of extreme importance to me that you have at least the science so that you can make a wise decision with what you do. So the question is, like, how will we use the technology we have? That's the question on the bottom. How will we use the technology we have? Just because we have the ability to do something, should we do it? Just because we have the ability to do something, should we do that? All right, and that lends to the very end here of part 2. I know I sprung some new math there on you, but I wanted to change things up a little bit, and I knew you could do it. All right, I'll see you in class. You're important, and that's it.